Coming in from outer space, a keen-eyed Martian trying to unravel the complexities of structure and function in a city might spot something subtle but nonetheless significant: a variety of building styles. And on a series of visits, perhaps over hundreds of years, the Martian might realize why. With time, some structures fall into decay, while new ones take their place.
Like cities, cells change with time. Of course, the timescale is quite different, and there's no real equivalent of architectural style. But bits and pieces inside cells do wear out and are replaced with similar structures. And there's another sort of change. Like cities, cells can grow. In fact, growth is a biological term. Stumbling across a site like this, the Martian might tie together the long-term changes seen in cities with the sort of things that go on here. And watching carefully, the Martian might begin to understand the rules or principles for how large structures in cities are assembled. Now, what we'd like to know is when cells assemble large structures, such as organelles from their component macromolecules, is there a similar set of rules or principles of assembly? Just consider a high eukaryote cell undergoing the cycle of growth and division, and concentrate on the nucleus.
Before cell division, in interphase, the DNA is replicated, and this DNA is assembled with histones and other proteins to form chromatin. Condensation of the chromatin produces chromosomes visible at the start of mitosis, in this case in a plant cell. The nuclear envelope then breaks down, ultimately allowing the two sets of chromosomes to reach opposite poles of the cell. And at the end of mitosis, a new plant cell wall and each set of chromosomes enclosed by a newly assembled nuclear envelope, finally giving two new interphase cells.
Now, with the assembly of chromatin in interphase and the assembly of two new nuclei at the end of mitosis, the nucleus provides an excellent example of assembly. An excellent example, but a very complex one. So to try and tease out any principles of assembly, we better start with some simpler systems.
These are viruses, and they're each composed of a sequence of DNA or RNA packaged in a protective protein coat, arranged in regular shapes. Classically, viruses have given useful insights into cellular processes. Dr. Joe Butler of the Medical Research Council has long experience in this field.
When a virus infects a cell, it takes over the whole of the cellular processes and suborns them to make more copies of the virus. This means that instead, for example, of having the nucleus and the nuclear DNA being replicated in 12-24 hours, you may get a situation where copies are being turned out extremely fast. Instead of 1 or 2 points where DNA synthesis is occurring, you will get hundreds or thousands of places within the same cell. Hence, you've got a tremendous concentration of the material of interest to you.
This is tobacco, and the variegated or mosaic patterning is the symptom of an infection, a viral infection. The virus responsible is called tobacco mosaic virus, or TMV. Seen through the electron microscope, TMV looks like this: thin rods, each about 300 nanometers long. When looked at more closely, the rod is seen to be a helix, made from just one type of protein subunit, repeated round and round 2,130 times. This protects a single strand of RNA, which spirals right along its length.
Here I've got a rather simpler model of TMV. Instead of actually showing the shape of the subunit, we're just using a little wedge as of cheese. And here you can see the RNA at the end. The important thing to take home about this model is that these subunits are all packed into a regular arrangement of helix spiraling downwards from the one end right through to come up at the other end. But how is it put together? Our knowledge on this really dates back to a very important experiment in 1955 done by Frankel, Conrad and Williams, where they took isolated RNA and the isolated protein subunits and showed that if they put these two together in the test tube, they were capable of self-assembling to give infectious virus. This was the first time that the process of self-assembly had been shown for any biological system in a laboratory setting. The question is, how can this and these isolated subunits get together to give a definite structure?
So what TMV reveals is an important biological concept, self-assembly. Just mix the components together and hey presto. No tools, no scaffold, no hands. All that's needed are the components and the final structure. The ultimate in DII, do it itself. But when we speak of biological self-assembly, do we literally mean that the components throw themselves together? Wouldn't we expect something a bit more sophisticated? For example, perhaps a defined sequence of assembly.
The protein subunits can start to aggregate and assemble together and put themselves into bigger and bigger aggregates in this sort of way. And ultimately, they come round to form this disc-like structure. This has two complete rings of subunits facing in the same direction and with a central hole and a gap like a pair of jaws between the two subunits onto that central hole. Now, what happens is we have a special region of the RNA with a rather special sequence here, shown in red, as a single-stranded loop on top of a rather weakly base-paired stem. And this loop inserts into the central hole of a disc, and the special sequence comes round into the pair of jaws, and it binds round. The binding interaction melts this hairpin loop, so that instead of just a small loop here, you get a complete turn of RNA inserted and bound between the two layers of the disc. Some feature of this binding interaction causes the disc to dislocate and spring apart from this disc structure into a lock washer. And these subunits come tightly together to give a complete, stable structure, so that we end up with a structure like this one, where the turn of RNA is now contained between these subunits, and it comes out the top and dives back down the central hole. Assembly can now continue by another disc coming in onto the top, with this loop of RNA now inserting between the two surfaces and allowing this disc to come in, bind onto that step there, and form additional subunits of the growing nucleoprotein helix.
The second disc, engaging with the RNA, it too springs open to form a lock washer, which then anneals with the first unit. Subsequent discs follow the same pattern, each binding with a continuing RNA loop. That this happens in practice is supported by electron micrographs of partially assembled TMV. The growing rods, each with two free tails of RNA trailing from the same end, are perfectly evident. Disc after disc is added until the long loop of RNA has been drawn right through. The short tail of RNA at the other end is also coated. A new infectious virus is formed. So self-assembly is clearly a powerful mechanism. But is it the whole answer?
Self-assembly is extremely important in biological systems, and particularly, of course, in viruses. But no, it isn't the whole answer. For a start, proteins may be involved in slowing down the process and stopping unwanted interactions. You can end up with chaperones, which stop two components coming together when they shouldn't be. Another thing is that as things are assembling, the next interacting site may only be generated as you complete one sub-assembly, so that only once this one is complete can the next one come in and interact with it. A classic example of this comes from a bacterial virus, the bacteriophage T4.
The three parts of the T4 bacteriophage, the tail fibers, the tail, and the head, all undergo defined sequences of pre-assembly before they come together. We'll concentrate on the head. The yellow proteins will form the mature head, but they require the pink proteins to provide a jig or scaffold for assembly. For this reason, the pink protein is called scaffold protein. Once formed, DNA enters this prohead and an enzyme gets to work degrading and so removing the scaffold protein. The prohead then assumes its mature shape, ready to assemble with the other components.
So there are our principles of biological assembly. Firstly, self-assembly. Secondly, the use of other factors, tools such as scaffold proteins and enzymes. And finally, a defined sequence of assembly. Now, the other thing to notice is that we didn't uncover these rules or principles by experiments on whole cells, but from experiments in test tubes by using cell-free systems. Now, can we take the principles and the exploitation of cell-free systems and apply them back to the much more complex problem of the nucleus?
Eggs, unfertilized frog eggs of the species Xenopus levis, packed with materials which will fit them for rapid development once fertilized. And for biologists here at the Cancer Research Campaign's laboratories in Cambridge, this means the source of a remarkable cell-free system for nuclear assembly. The eggs are degelid and the bad ones removed. The best of the batch are then transferred for centrifugation. Spinning the eggs gently crushes them, and a further and more vigorous spinning stage separates the cellular components. The result is 3 distinct layers, and it's the middle layer that's of interest, because this contains all the components needed for nuclear assembly except for DNA. There is no DNA in this fraction.
The most remarkable feature of the cell-free system derived from this material is that when DNA is added to the egg extract, it can be assembled into structures which look exactly like conventional nuclei. We call them pseudonuclei, but so far as we can judge by structural and functional criteria, the DNA has been assembled back into structures which show the remarkable complexity of a cell nucleus of the sort that you would find in an authentic cell.
What then are the main structures in a nucleus? Well, it has an outer envelope peppered with nuclear pore complexes. These only allow specific large molecules in, and other equally specific large molecules, like messenger RNA, to pass out. Seen here under the electron microscope, the pores are more than mere holes. They are complex structures which act as highly selective gates. The nuclear envelope itself consists of 2 membranes, and inside is the chromatin, the complex of DNA and proteins which, when condensed, forms the chromosomes visible at cell division. But how are the pseudonuclei assembled in a cell-free system? Do any of the rules we gleaned earlier come into play? We'll start with the chromatin.
The first thing that happens to naked DNA when it's added to the cell-free system is that it becomes assembled into chromatin. Now, there are several levels of chromatin structure which can be reassembled, but the one that we've studied most intensely is the assembly of the nucleosome subunits of chromatin.
At its lowest level of packing, chromatin exists as a molecule of DNA wound around histone complexes at regular intervals to give what are called nucleosome subunits. This gives the appearance of a beaded string.
Here I've got a model of the histone octamer, which forms the basis of that structure. To each of the 4 core histones, colored here in the different colors shown on this model. And an essential feature of nucleosome assembly is that DNA must interact with the histones to produce this subunit structure with the DNA wrapped twice around the histone octamer. Now, in assembling such structures, there are two key problems. One is that the basic charges of the histones repel each other, so bringing 8 of them together is a challenge. And secondly, it must prevent non-specific interactions because histones will bind to DNA at any point on that DNA. And something has to prevent that from occurring so that you get a regularly repeating structure. Now, we can do this by self-assembly if we use high salt concentrations, for example, but it's a slow process and a difficult one. The cell does it a different way. The cell uses assembly factors which mediate those interactions, for example, by binding to histones and masking their positive charges, and therefore allowing the DNA to interact with them in a much more ordered fashion. And this role of assembly factors led us to coin the term molecular chaperones to describe this interaction in which the non-specific interactions are filtered out and only the correct assembly pathway is allowed to occur.
The beaded string is only the first level of folding. What happens beyond this is far from certain, but the chromatin must be further compacted to give the highly condensed chromosomes seen at mitosis. The chromosomes are further arranged in distinct configurations, something observed as long ago as 1885 by the Austrian biologist Karl Rabel. But what wasn't evident was whether chromosomes and their arrangement existed in the interphase nucleus, or whether in interphase the chromosomes decondensed to give what might be termed an unordered soup. Recent evidence from natural nuclei is providing the answer.
Structure of the nucleus is very highly ordered, and it differs fundamentally from the widespread concept that it's really just a bag of spaghetti. In fact, there's a precise structural architecture throughout the cell nucleus, which plays an important part in its function. The clue that there was originally a structure inside came as long ago as the last century from work of Rabel, who showed that when chromosomes divide, they occupy a particular configuration at the end of division. And there's now further evidence, notably from the work of Sadat, to show that this configuration is continued through interphase. So it doesn't just disappear at the end of mitosis, but chromosomes hold their station after the end of mitosis and as the interphase nucleus forms. So that's one clear line of evidence that there is a precise ordered structure inside the nucleus. There are others. And one of the most striking of these comes from studies by Singer, who's been looking at the export of messenger RNA from its sites of synthesis on the DNA encoding that message out to the cytoplasm.
Robert Singer and his co-workers stain isolated nuclei with a fluorescent probe against certain specific messenger RNAs. When viewed under ultraviolet light, the probe makes messenger RNA show up as yellow.
What you might expect if the message is being made on the gene at a precise position is that that message might then diffuse away as a diffuse halo away from its site of synthesis. That's not what's observed. Strikingly, the message appears to go in almost straight lines from its site of synthesis out to the cytoplasm. So it's not just diffusing around, it's being targeted out along a discrete route, rather like tram lines within the nucleus. Now we have a third line of evidence that the nuclear architecture is precisely ordered. And this, interestingly, is a line of evidence that we can reconstruct in the test tube. And this concerns patterns of DNA synthesis.
DNA synthesis is studied at Cambridge by using the technique of fluorescent microscopy, as we saw when visualizing messenger RNA, but now coupled to the confocal microscope. The confocal microscope is based on an adaptation of light microscopy. It uses a scanning laser combined with an electronic imaging system. The effect is to allow planes or optical slices to be viewed within the specimen, much as if it had been physically sectioned. And the fluorescent staining produces these bright red spots at the sites where DNA synthesis is taking place, in this case in a reassembled nucleus.
We can observe that DNA synthesis is not diffuse throughout the whole of the nucleus, but is occurring at quite discrete sites. The striking thing about nuclei which we've reassembled from purified DNA is that a very similar pattern is reconstructed, not only from purified DNA from an animal cell, but purified DNA from bacteriophages, bacterial viruses which have never been organized in this pattern at any stage in their life history. The cell-free system can reimpose this same highly ordered structural pattern on DNA, on nuclei assembled from DNA of a bacteriophage.
So, given DNA, not only does the cell-free system assemble it into chromatin, but it further assembles a complete, apparently normal nucleus. Well, we've looked at chromatin, but what do we know about assembly of the other parts of the nucleus? In the cell-free system, DNA is first assembled into chromatin. Pore complex proteins bind onto its surface where they interact with one another to form immature pore complexes. Membrane vesicles also bind onto the chromatin, and owing to their lipid fluid properties, they spread and fuse, filling the spaces around the pore complexes. The final result is a continuous double-layered envelope with pore complexes firmly embedded within it, acting a bit like rivets, holding the two layers together. This can be seen in practice from an electron micrograph of a reconstituted nucleus. So, given the clear success of cell-free systems to construct fully functioning nuclei, what conclusions can we draw?
Studies of assembly of the cell nucleus in cell-free systems have led us to several conclusions. Firstly, we can see that there are definite pathways. It's not a free-for-all, but certain assembly steps take place before others, and we have a defined order of assembly interactions. Secondly, we can conclude that some of these assembly processes are mediated by specific assembly factors. We suspect that others will be self-assembly processes. For example, the fusion of membrane vesicles to form the nuclear membrane may be in part self-assembly. But we can certainly say that factors are involved in some steps, and at this stage we can suspect that they won't be involved in all steps. But we know there's a third level of regulation of assembly which we can study, and that is enzyme-mediated structural changes. For example, when the nucleus breaks down at cell division, certain proteins become highly phosphorylated, and they appear to play a causal role in nuclear disassembly. Conversely, when the nucleus reforms at the end of mitosis, the phosphate groups are removed, and the nuclear structure then reassembles. But to conclude, we can say that cell-free systems have allowed us to draw many conclusions, but they also need to be interpreted with caution. We can show that these things do occur in a cell-free system, but we need to be cautious before we assume that that's exactly how it happens in the intact cell. Nevertheless, in spite of that caveat, the cell-free systems offer us powerful experimental tools to study assembly mechanisms, and in addition, they allow us a completely new approach to studies of the function of the cell nucleus.
So we're beginning to understand the assembly of the nucleus and other organelles. But ultimately, we'd like to know how a cell builds a cell.